Hello friend, in this particular module, we are going to see different types of bioequivalence studies and bioavailability studies, including in vitro bioequivalence studies and clinical endpoint studies. Before going into the types of studies, we'll understand what is crossover and how it is randomly assigned for the study. So, a type of clinical trial in which all participants receive the same two or more treatments, but the order in which they receive them depends on the group to which they are randomly assigned. What does it mean? So, for example, you are going to do a two-way crossover study where test product and reference product is there. So for a subject who has assigned first test followed by the reference, the test product, which number he will be getting, that will be randomly assigned. For example, the first period, there are 30 subjects, 15 will be assigned to test, 15 will be assigned to reference. Sequence is test followed by reference. He will be assigned to the test group, but which subject number, that will be randomly assigned by the randomization schedule. So this is what explained in the first paragraph. And now coming to the crossover trials. Crossover trials allow the response of a subject to treatment A to be contrasted with the same subject's response to treatment B. That means I am the person who is taking the test product, then I have to expose to the same treatment. That means the treatment B, that is the reference product. Also, how I will behave for both the treatments will be understood. Hereby, we are removing the patient variations. So, removing patient variation in this way makes crossover trials potentially more efficient than similar sized parallel group trials in which each subject is exposed to only one treatment. Parallel group, for example, 30 subjects will be there in the study and 15 will be assigned to test, 15 will be assigned to reference. That's all. There won't be any crossover, so that the intersubject variability only we can understand. The intrasubject variability won't be there, and these kind of trials which need more power, that means more sample size to conclude bioequivalence. In case of bioequivalence studies, the widely used design is a two-way crossover design. Sometimes we use three-way crossover design, parallel study designs. Three-way crossover generally used in the pilot stage or the development stage of the product, where you want to test two test products against the reference product. So here the subject will be exposed to three different treatments. That means the test one, then test two, and the reference in a randomly assigned sequence as well as the randomly assigned group. So this is how the three-way crossover studies will generally be carried out. And in case of replicate designs also, this three-way crossover will be adopted. In case of reference replicate, where to understand the reference to reference product variability, reference product will be administered two times and test product one time. So the sequence will be RRT or TRR, like that will be there. So that also will be done in a three-way crossover manner. Then the parallel study designs are generally used for long washout drugs, and this kind of study will be generally adopted for the clinical endpoints studies also. So parallel study designs are very limited in bioequivalence and very rarely adopted because you need more sample size and you are unsure of bioequivalence conclusion. And then finally, we have replicate design studies where three-way as well as four-way replicate designs are used for the highly variable drugs, where you can determine the variability of the reference product based on which the test product bioequivalence assessment decision will be taken considering the variability of the reference product. So this is where the highly variable drugs comes into the picture. The bioequivalence studies generally adopts the open label design, balanced randomized. That means when you randomize first period, 30 subjects are there, 15 will be test, 15 will be reference, like that balanced randomized. Then it can be a three-way, two-way, four-way crossover study, and depends on the crossover, the number of treatments will be there. Either it can be two or three treatment studies, and the test product and one RLD or vice versa will be used in case of the three treatment studies. And there will be sequence where three sequence, two sequence studies. Generally, it is two sequence studies, but sometimes three sequence studies and four sequence studies also will be adopted. Based on the dosing, we can define different types of bioequivalent studies. So we can say that if you use only one dose, that is single dose fasting study, if you compare the single dose pharmacokinetics of the product. So that is called single dose study. So this will be classified into further fasting study and fed study, where you administer under the fasting condition. In case of fed study, you after the breakfast you administer the single dose of the test product or the reference product. And then we have multiple dose fasting study, multiple dose fed study. So here we assess the steady state pharmacokinetics of the product under fasting condition or fed condition. This condition, fasting or fed, will be decided based on the actual drug product treatment. For example, in case of diabetic, anti-diabetic drugs, metformin and all, it is given under the after food. So that generally adopt for the fed condition. In most of the drugs which it doesn't have any impact of food on the pharmacokinetics will be done on the fasting state. So here, till the pharmacokinetics achieved the steady state, the drugs will be given for 5 days or 7 days, depends on the dosage regimen, 2 times a day or 3 times a day or single time a day. So then the pharmacokinetics will be, samples will be taken after the steady state achievement after 5 days or 7 days like that. So then the pharmacokinetic parameters will be characterized. Then there will be food effect study, as I earlier explained in some other module, where the for one product, given product, the fasting pharmacokinetics and fed pharmacokinetics. After food, what is the pharmacokinetics? That will be established, and the food effect will be established on the pharmacokinetics. Then finally, the replicate design study, which I already explained for the highly variable drugs. Here is the multiple dose studies. So I explained in the previous slide, it is used for the assess the steady state pharmacokinetics. Dose will be given for 3 or 5 or 7 days based on the dosage regimen and the reported steady state achievement. So depends on the half-life and accumulation. The steady state will be achieved in 3 or 5 or 7 days. So after steady state achievement, the pharmacokinetics blood samples will be taken for the duration of the dosage regime. That means if it is twice daily means 12 hours the pharmacokinetics samples will be taken. If it is once daily means 24 hours. Thrice daily means for 8 hours the sampling will be taken and the pharmacokinetic parameters will be calculated. So this is fast and fed based on the product label. I given you example of metformin. Then steady state achieved after specified time. This I already explained. And then the parameters like steady state maximum concentration, steady state minimum concentration, and AUC 0 to dosing interval. So dosing interval means if it is twice a daily means 0 to 12 hours. That sampling time points will be 0 to 12 hours. So we will calculate the AUC 0 to tau, that is dosing interval, and C tau, that is the last concentration of the dosing interval. So if at 12 hour, what is the concentration that is called C tau at the 12th hour of the particular dose. Then the fluctuation index, all these parameters will be calculated during the multiple dose studies. Then finally, the bioequivalence criteria applied for the CSS maximum concentration, minimum concentration or C tau and AUC 0 to dosing interval 0 to tau. So depends on the regulatory submission, either it is US FDA or Europe. Based on the recommendations by the guidances, we will apply the bioequivalence criteria. So here the type of studies required for each and every Western regulatory market as well as different market like South Africa, Canada, TGA are just presented for the reference. Like for US FDA, we need generally single dose fasting and fed studies for most of the products. And for Europe, you need fasting studies only for the immediate release product and fast and fed studies for the delayed release and extended release products, and multiple dose studies also required for extended release and modified release products. Then for South Africa, Canada, Australia, generally required fasting studies for the immediate release formulation. TGA, like Australian regulatory, they follow the European guidelines, and Canada also follows the European guidance and also refers some of the products US FDA guidance. Then in case of cytotoxic drugs like cancer drugs, we need to use patients for the study population. Sometimes the bioequivalence studies are designed based on the clinical endpoint. So those drugs which doesn't have absorption in the gastrointestinal tract. So for those drugs, we design clinical endpoint studies. Some drugs which are locally acting in the gastrointestinal tract, like phosphate binders, bile acid binders, and some drugs which is not quantifiable. There is no quantifiable plasma concentration. For those drugs also we do clinical endpoint studies. And also Locally act drugs for locally acting means there are ointments, gels also we do the clinical endpoint studies. So here is the example: sucralfate, which doesn't have any absorption; rifaximin doesn't have absorption. These are locally acting. And ointments, gels, creams, this all, for all these products we go for the clinical endpoint, where the efficacy of the product are measured and statistically compared. Sometimes the bioequivalents are being done with the use of in vitro methods. So the in vitro studies are useful in case of those drugs which are not absorbed in the gastrointestinal tract and locally acting with respect to some mechanism like phosphate binding, bile acid binding, and also albumin binding. Classical examples like cholestyramine, sevelamer carbonate, lanthanum carbonate, calcium carbonate. So these are the molecule which doesn't have any absorption in the gastrointestinal tract, and the how fast the binding happen and what extent the binding happen will be compared. Like bioavailability, the rate and the extent of the absorption of the drug into the systemic circulation. Here, the rate and the extent of the binding to what extent it is happening. If it is comparable for both test and reference product, that will be used for the bioequivalence assessment. So the binding constant will be established, and these kind of studies follows the bioequivalence principles and the bioanalytical method validation protocols, and generally considered as in vitro bioequivalence studies. In this slide, let us understand what is nonlinear pharmacokinetics. So generally, if you have a Cmax of 30 nanogram per ml for a 100 mg dose, then for the 200 mg dose, you will have 60 nanogram per ml approximately. 60 nanogram per ml the Cmax, and if it is 300 mg, it is expected the 90 nanogram per ml is the dose. This is called linear pharmacokinetics. Similarly for the AUC, but some drugs doesn't work like this. They do have different pharmacokinetics of different strengths. For example, 100 mg may not be doubled when you dose 200 mg, where you will get only 40 mg or 45 mg. So this is called nonlinear pharmacokinetics. So generally, when there is different strengths are used for marketing, so for example, 30 mg or 60 mg, two strengths are used. Set 60 mg, you do the bioequivalence study, and 30 mg you will get the bioequivalence based on the dissolution properties and in vitro properties. But in case of nonlinear pharmacokinetics, you have to do bioequivalence studies are both the strengths. So sometimes if there are three strengths, you have to do the higher strength and the lower strength, middle strength, you get the bioequivalence. So sometimes in case of nonlinear pharmacokinetics, we have to do the multiple those studies. So this is how the nonlinear pharmacokinetics influence the choice of bioequivalence studies. Now, what happens if a biostudy fails? Why the biostudy fails? How the biostudy fails? If the test product is lower or higher measures of the rate and extent of absorption. So the Cmax is low, or AUC is comparatively low for the test product, or it is very high, then the study product will fail for the bioequivalence. In that case, you have to see that whether you have to modify the formulation to match with the reference product. So sometimes the bioequivalence study fails because of the poor design of the bioequivalence study, where the drug is more variable and you have gone for less number of subjects comparatively for the study design. So in that case also, study sometimes fail. In that case, you have to repeat the entire study with the higher sample size. In case of higher plasma levels obtained for the test product, what will happen? The safety of the for product will be questioned. So there will be higher plasma concentration, so you tend to get some kind of adverse events for the product. In case of lower plasma level of the test product, there won't be any efficacy. You will have poor efficacy, so that cannot be used. In case of variability, also the test product, higher variability, generally it is a drug property rather than a drug product property, but sometimes drug product can influence the variability. That time also the bioequivalence study fails. So there is a bio waiver system where some products you can get the bio waiver. You need not do any bioequivalence study. You can directly apply the dossier along with the in vitro data. It depends on the regulatory agencies and the recommendations provided by the regulatory agencies. So generally, in case of class 1 drugs, that is highly soluble, highly permeable drugs, biowaiver are considered, and the solution dosage forms biowaivers are considered. Injectable solutions also biowaiver considered, and biowaiver considered in case if you do the higher strength for the dosage form, then you can get the lower strength biowaiver. So these are the biowaivers examples adopted in the bioequivalence study system. So, in this module, we have seen briefly about the bioequivalence study types and designs. Now, going forward, we will be seeing the different phases of this bioequivalence studies, which have been briefly explained earlier. Now, we are going to see in detailed manner. Thank you.